Hello, we are from CEEC 2226B2A, group number 2, and we are going to do a brief presentation for our Environmental Engineering Laboratory mini project. The objectives of this mini project is to propose an experimental procedure to treat a water sample with high turbidity and total solids. Next, removing or reducing contaminants to make the water suitable for its intended use. Lastly, ensuring alignment with sustainable water management goals such as SDG Objective Number 6, Clean Water and Sanitation, and SDG Objective 14, Sustainable Use of Aquatic Resources. For this mini project, we are going to conduct 3 tests. Number 1, physical test. Number 2, chemical test. And number 3, biological test. For the physical test, we are going to conduct 4 tests, which are turbidity test, TDS, conductivity, and also volatile suspended solid. Next, the second test is chemical test, which are 3 tests conducted: nitrate, nitrite, and COD. And for the biological test, we are going to conduct BOD and also E. coli and total coliform test. Before carrying out the test, we had to dilute our water because it was too contaminated. The ratio we used was 1:10 ratio. The first experiment we are going to conduct is turbidity test. First, fill sample into sample cell and then wipe before putting into turbidity machine. And then, press auto range until auto range appears on the screen and press read. Wait until reading is stable and read the reading. Next, the second experiment is conductivity test and total dissolved solid. For these 2 experiments, they are both carried out the same way. Firstly, wash probe with distilled water and put into the sample. Next, press COND or TDS and then press read. Wait until reading is stable and log appears on screen. And then finally record the reading. After that, take out the probe and wash with distilled water before pressing exit. Lastly, the final physical test, which is volatile suspended solid. For this test, you have to weigh the empty crucible first. After weighing, you have to put in the sample into the crucible, and then you have to weigh again the sample with the crucible. After weighing, you have to put the crucible inside a preheated oven for approximately 6 hours. After 6 hours, transfer the crucible into a furnace for 30 minutes. Then after 30 minutes, let the crucible cool down in a desiccator and then you can take the reading. Next is chemical test. The next test we are going to carrying out is nitrite test. For nitrite test, first you have to fill a square sample with 10 ml of sample water. Next, after filling the square sample, you have to add the nitrite reagent powder and then you have to swirl to dissolve. A pink color will develop if nitrite is present. And then, after swirling, let it react for 20 minutes. In the meantime, we can prepare for nitrate test. Fill a mixing cylinder with 15 ml of water sample. And then, add the reagent and shake for 3 minutes. After 3 minutes has passed, a 2-minute reaction period will begin. After 2 minutes, carefully pour 10 ml of sample into clean square sample cell. And then you can add a nitrate reagent powder pillow to the sample cell. And then cap the sample cell and shake for 30 seconds. After that, let it rest for 15 minutes. In the meantime, the nitrite sample is ready to be tested. First, you have to enter the program number for the nitrite testing. And then, you have to wipe the sample cell cleanly using a tissue to avoid any fingerprints. And then insert the sample cell into the spectrophotometer. This is the blank result. And then you take out the blank and then repeat the same step for the sample cell. When nitrite is done testing, you can test for the nitrate test. Repeat the same step as before. Next, chemical oxygen demand test, COD. Firstly, the blank sample for the experiment by adding 2 ml of distilled water and COD high range reagent by slooping the test tube about 45 degrees. Then carefully invert the test tube so that the reagent can do its work. Then use a micropipette to drop 2 ml of sample water into the test tube. Then the sample will be undergoing heating process for around 6 hours. After heating for 6 hours, the test tube will be cooled down first before being tested in the spectrophotometer. Next, the first experiment for the biological test, which is the totocoliform and E. coli test. First, you have to pour 100 ml of water into the vessel and add reagent. And then, close and shake until the reagent dissolves. After that, pour the sample into quantity tray and seal. After sealing the quantity tray, place the quantity tray into the incubator for 24 hours. Next, after 24 hours, the quantity tray is scanned using a fluorescent light. As you can see, the neon color indicates the availability of the E. coli. Next is BOD test. First, you have to prepare the dilution water by adding 1 liter of distilled water into the dilution water container, and then followed by adding 1 milliliter each of chemical reagents, which are phosphate buffer solution, calcium chloride solution, magnesium sulfate solution, and ferric solution. After that, let it aerate for 1 hour. Next, fill 50 ml of sample into a BOD bottle and then fill the BOD bottle with dilution water to its neck. Using the ohmmeter, take the reading. After that, store it into the incubator for 5 days. After 5 days, repeat the step and take the reading. So now we are going to conduct the jar test. First, prepare coagulant and sample. Prepare the coagulant at different volume but for the same concentration. For example, 2 ml, 4 ml, 6 ml, 8 and 10. Start the process and immediately put all the coagulant into the beaker. The timer is set for 2 minutes at 140 revolution per minute RPM. For the next process is the flocculation process. Set the timer for 5 minutes at 70 RPM. Start the machine and let the process begin. After that is the sedimentation process. Here is the condition after carrying out jar test. After that, we repeated the same experiments using the cleanest water, which was with the 10 ml coagulant. Here are the results that we obtained after carrying out the experiment.